Hi, everyone. My name is Patrick Menke. I am a current undergraduate student at Bradley University, studying under my mentor, Dr. Jen Jost. I'm also in the biology department's 4+1 program and hope to obtain my master's degree in the spring of 2024. And today I wanted to present some of my preliminary work that I've done concerning the effects of potassium chloride on zebra mussels. So first, I just wanted to start with some quick background information on what zebra mussels are. So zebra mussels, or Dreissena polymorpha, are an invasive bivalve that were originally native to parts of Eurasia, like the Caspian and Black Sea. They first spread into Europe and then came into America in the late 1980s through ballast water discharge. Since their introduction in the late 1980s, they have shown to cause great economic and ecological damage. In terms of economic damage, they have a abyssal thread that allows them to attach to hard substrates. This causes them to clog certain water intake pipes for facilities like electric generating plants and water treatment facilities. They reach high densities very quickly, so this clogs the pipe, forcing these companies to manually remove them, which can be quite expensive. Some studies have shown that since their introduction into the mid-2000s, they've been responsible for anywhere between 160 million and 460 million dollars in damage. In terms of ecological damage, these mussels are able to reproduce at a very rapid rate. A female mussel can lay anywhere between 30,000 and a million eggs in a season, and they have the ability to spawn multiple times in a spawning season. They also have a free swimming veliger stage, which allows these young to have a better chance of survival and being able to turn into reproducing adults. Along with this, their abyssal thread again allows them to connect to native mussel species, which can harm these mussels since they compete for similar food resources, and they can also outcompete them for space. These mussels have also shown that they're able to survive brief periods of aerial exposure at a variety of different temperatures. Some studies are even showing that these mussels can survive periods of exposure for upwards of 5 days. This is concerning as it could potentially allow them to travel on land by attaching to watercraft on their abyssal threads and infesting new bodies of water and establishing new populations. While there has been some work done at looking at different lethal chemicals and substances to control their spread, such as potassium chloride, Zequinox, and EarthTech QZ, many of these pose the same difficulty of it being almost very hard to maintain the specific lethal concentration in an aquatic environment. And so for my study, the overall objective is to look at how we can use one of these lethal chemicals, potassium chloride, during aerial exposure to hopefully inhibit their on-land travel. So on my pictures on the right, I just wanted to show some brief examples of what zebra mussels look like. So these pictures were taken by me. These are mussels we've hand collected from Banner Marsh. This is where all of our mussels are collected from. So as you can see on the top picture, that's just a representative image of a larger zebra mussel that has smaller zebra mussels attached to them. In the bottom photo, you can see just kind of our array of zebra mussels and how we randomly assign them to different treatment groups to avoid bias. So the objectives of some of this preliminary work was to first determine the lethal potassium chloride concentration, both in air and in water, how this lethal potassium chloride concentration can impact native species, and then how aerial temperatures can impact mussel mortality when exposed to that lethal potassium chloride concentration. So for the first objective, to observe the lethal concentration in water, we hand collected our zebra mussels from Bell's Landing within the Banner Marsh State Fish and Wildlife Area, and then we transported them back to Bradley in insulated containers with river water collected from the site. Once back at Bradley, mussels were randomly assigned to various potassium chloride concentrations. They were housed in 2-liter containers that each contained 5 mussels. We had an N of 3 for each treatment, and then we had checked survival every 12 hours. So as you can see on this figure on the right, these are our percent survival after the 72 hours for the different potassium chloride concentrations that we tested. So we determined that the lethal concentration is somewhere between 125 and 150 milligrams per liter to cause complete mortality within the 72 hours. Again, continuing with this first objective, in a similar fashion, to determine the lethal concentration in air, we've again collected zebra mussels from the same location at Bell's Landing in the Banner Marsh State Fish and Wildlife Area and transported them back to Bradley in a similar fashion. They were again randomly assigned to different potassium chloride concentrations and housed in the same 2-liter containers, again with 5 mussels per container. The difference is that since they were in air, we had to spray the potassium chloride solution onto the mussel. Each jar got the same amount of solution sprayed, and the only thing that changed was that each spray was a different potassium chloride concentration. Our mussels were then exposed to air in what we found were to be non-stressful aerial conditions for 72 hours, and then moved to an aquatic 24-hour recovery. Survival was again checked every 12 hours, and then after the 24-hour recovery period. The non-stressful aerial conditions were determined by, we found no mortality within the 72 hours, and they were 15 degrees Celsius and 75% relative humidity. I just wanted to show a picture on the right. This is an example photo of one of our environmental chambers here at Bradley University. These allow us to control different aerial conditions such as temperature, relative humidity, different types of lighting, and a few of them can even change the carbon dioxide concentration in the air. And so the results from this, again, on this graph or on this figure, you can see the percent survival and then the different potassium chloride concentrations that we tested. We originally started lower, assuming that it would be near what we had found in water, as in aerial exposure is more stressful in these mussels, but we quickly found that this dose needed to be much higher, and we assume that this is most likely because the mussels are not actively filtering the solution when they're exposed to air. And so we were able to determine that that lethal concentration was between 350 and 400 milligrams per liter. And then for our objective 2, we decided to pick just a one native species to see how they reacted to that lethal potassium chloride concentration in water. And so to do this, we chose the northern crayfish, or Phexonus virilis. And as you can see in the photos on the right from the USGS, the Phexonus virilis has a native range that overlaps greatly with a lot of the invasive spread of zebra mussels. So the zebra mussels, again, were collected in a similar fashion, and the northern crayfish were purchased from Carolina Biologica. The crayfish were then also randomly assigned to one of 2 potassium chloride concentrations, either 0 or 125 milligrams per liter, and then at 2 different water temperatures of 15 or 25 degrees Celsius. We checked survival every 12 hours for 72 hours, and then after the 72 hours, we measured their writing response and their lactate concentration. We measured their writing response as if the crayfish were affected by the potassium chloride and they were not able to right themselves, they would lead them open to more predation and effectively causing mortality. We measured lactate as it was a simple way to show if the crayfish were experiencing any physiological stress due to the potassium chloride. And so as the results from this, you can see on the graph on the left, there was no significant differences in the lactate concentration for crayfish exposed to 0 and the 125 milligrams per liter of potassium chloride. And then there was also no differences between the 2 different temperatures of 15 and 25 degrees Celsius. And then here on the right, you can see on the top of the figure A, or 4A, that was the aerial reaction time. And then on figure 4B, that was the aquatic reaction time. And we again did not find any significant differences between the different potassium chloride concentrations. And we did measure this in water and in air. One for water is that's where they mostly live. And then in air, we wanted to measure in air too, just as if the crayfish were experiencing a stressor in the water, they may leave the water for shorter periods of times. And we wanted to see if they did escape the water to leave the stressor, if that would impact their aerial writing response. And then for the third objective, we wanted to look at how different aerial temperatures could impact mussel mortality when exposed to that optimal potassium chloride concentration. So again, mussels were hand collected and then assigned to both various temperatures and to 2 potassium chloride concentrations. So that optimal dose of 400 milligrams per liter or a control of 0 milligrams per liter. They were assigned to either 15 degrees Celsius, 20 degrees Celsius, or 25 degrees Celsius, and all at 75% relative humidity. We again check survival every 12 hours with another 24-hour aquatic recovery. I wanted to show this photo on the right. This photo isn't directly from this study that we did. It's just a representative image of what the mussels were housed in. So as you can see in the photo, those are the 2-liter containers we used, and there were 5 mussels within each container. And then they would have been sprayed with that optimal potassium chloride concentration. And so here are the results from this third objective. So as you can see in all three aerial temperatures, mussels exposed to that lethal concentration of potassium chloride experienced a faster mortality compared to mussels that weren't exposed to potassium chloride. One interesting thing we did find was that when we had restudied this objective, the first temperature of 15 degrees Celsius was the same treatment that we had used for mussels when we were trying to discover the optimal lethal concentration of potassium chloride. And when we had repeated it for this study, we found that they didn't have complete mortality, which makes us want to have to re-look at this and potentially increase that optimal KCl concentration. And then so some of the future work that needs to be done, and some of this has already been started, it's just continuing. So first, we want to determine the effects of post-exposure water conditions on long-term growth. And so to do this, we will be exposing mussels to a sublethal potassium chloride concentration, and then having a 72-hour aerial exposure, and then resubmerging them into different water conditions for 4 weeks to see if it can impact their long-term growth. So some of these post-exposure water conditions include like water temperature, food quality, or even food quantity. And we have already started this at looking at different water conditions, or at water temperature, sorry. But the mussels are still growing, so we don't have the results for this, but you can see on the image in the bottom right, that's a picture of the mussels right before their aerial exposure. For this, we had to use smaller containers as the post-exposure water temperatures are controlled by chillers in our lab, and we had a limited access to those. And so we had to fit more jars in each chiller. And so to do this, we had to create smaller containers for them and then closely monitor the different water conditions. And then another thing what we're working on currently is to determine the levels of different stress indicators when mussels are exposed to potassium chloride in aerial conditions. So currently I'm working on finishing some of the baseline indicator work for mussels that aren't exposed to potassium chloride in aerial conditions. This will just give us a sense to see how potassium chloride specifically can impact these stress indicators. This is not relatively well studied in zebra mussels, even in aquatic conditions, let alone aerial conditions. So this is a big undertaking that will have to be done before I can move on to looking at how these stress indicators change when exposed to both potassium chloride and aerial conditions. And then another thing that we want to look at in the future is how when mussels are resubmerged into water, if the potassium chloride that is sprayed onto them during exposure can impact the potassium chloride concentrations in the bodies of water themselves. This is important as if it does change the potassium chloride concentrations in bodies of water after resubmergence, it could potentially impact the native species that are found there and could potentially harm them more than the zebra mussels are harming themselves. So in order to do this, my lab just recently acquired a meter that can actually measure the potassium concentration. And so that's something we're upcoming this semester that we are working on. And then just some quick acknowledgements of who helped make some of this preliminary work possible and for some of my future master's thesis work. So first, I'd just like to thank the Bradley University Biology Department for allowing the use of their growth chambers and for funding some of parts of the study. The Bjorklund Endowment Fund, who sponsored the purchase of the crayfish and the materials needed for the lactate concentration measurements. And then also the Bradley University Liberal Arts and Science Undergraduate Summer Research and Artistry Fellowship Program, which allowed me to be able to spend my summer the summer of 2022 in lab the whole summer. So I was able to do some of this preliminary work before entering the graduate program here at Bradley. And then also the Illinois Department of Natural Resources for the collection permits that were used in order to collect zebra mussels from Banner Marsh. Thank you again for inviting me here to speak at the Emma Kwan Symposium. As this is preliminary work and I'm heading into my graduate work, I appreciate any feedback or any support for this upcoming project or any input that may be helpful in the future. Again, thank you so much and have a good rest of your afternoon.